right? Do I agree with that? The goal of BIDS,
everyone, I am going to record to share with the people that are not there. So I forgot to say this. So sorry, I am recording now.
Is to make more data accessible, right? So the whole point of sharing is to share for yourself in 6 months with people in the lab, with other researchers in the world. Now, if we look at the current spec, what we have about PET's derivative is that common output, mostly of pipelines. That's the idea, capturing the data and the metadata. And again, so that it's human and machine readable, same as before. So I would say that the goal of this derivative is still the same, is still to make more data accessible, right? So the goal hasn't changed, whether it's raw data or derivatives, with yourself, with people in the lab, with other researchers. So now the last things I want to say is that it means that, as it states in the current spec, it means we need to provide information on how this was derived, right? That's the whole point of derivatives. And for this, there is two main approaches. So the first approach is basically the JSON, and of course the final files, which is specifying something like this was derived using pipeline XXX, see the code, couple of stuff about the parameters, the main aspect of it, right? So you can still reproduce, understand everything, because you put just one giant JSON and the code that goes with it. And the other approach is to do step one, here's the JSON, the file that goes with it, which describes what you've done, step two, step three, and so on. In my opinion, the most important in a meeting where we don't want to spend too much time is to agree on the final output, but last step, because this is at the end what we want to share, whether we share it with the first approach, here's the pipeline and the code, or we share it through a full description of everything. And the last thing is the elephant in the room, which could be file format depending on what we want to share. Right. So here, what Martin, for instance, has done is nice because the format doesn't change much. It's format that we agreed about already with TSV, JSONs, and Nifty. So that's relatively easy. And that's pretty much it. Thanks.
Great. Thank you, Cyril. Then this is very short intro to kind of BIDS and the state of the BIDS derivatives. And now Martin will take over and actually show you more PET-related work.
I will, thank you. And you can all hear me?
Yeah.
Great. All right, so I'll dig a little bit more into the current state of the PET-BID derivatives. But before I get started, I just want to say welcome to all of you, and welcome back to many of you. It's good to see both some old faces and some new faces, some faces that I've never seen before, but have contributed to this important work that we published last year on PET-BIDS. So good to see you all again. Just to give you a short summary of what we did with this work, was basically to try to standardize the raw data of PET. So as it goes out of the scanner, and then how we can store it. And the way that it turned out was that you had this dataset description and displaying the different versions and licenses. You could have some participant information. Then you would have some anatomical information where you would have the T1 weighted image, for example, and a corresponding JSON file with the metadata inside it. And then for the PET data, you would also have a JSON file with all the manufacturer information, units, tracer name, so forth. And then the corresponding PET data located in a Nifty file here, where you would often have it, depending on the data, either as a 3D image or as a 4D image. Then you could also have blood data, and that could have been recorded manually. And you could store that also in a JSON file with the metadata, and then a TSV file where you would have all the plasma data, whole blood data, metabolite parent fraction, and so forth. And this is really nice because it fits very well into some of the efforts that we're trying to do in terms of both sharing and reusing data. So this was a paper that was published by Gitte Knudsen and Bob Innes back in 2020 with many senior PET people. And this figure was included in the paper, basically describing this workflow where you would have the acquired data. That is what we in BIDS would call the raw data, or you could convert it into data to format for sharing. That would be the PET BIDS raw, as I just explained to you about. Then we would be able to publish this data, as we're already doing now, on a platform like Nørgaard. And then users that are interested in using this data, they can go and extract it. And then they have to do some sort of pre-processing and modeling to make this data useful. And that is where the PET BIDS derivatives comes in. And there are all these steps that you do to the images, image processing, blood processing, kinetic modeling, and that is what we will be talking about today. And the main goal is that you get these standardized outputs that you can then feed back into the platform, so others again can make use of this process data. And why is this important? I'll give you an example here. It could be many examples, but often it's quite difficult when you go out and read a paper. It's quite difficult to necessarily understand what has happened to the data. And I'll just give you an example here for two PET tracers from two different groups. And when you dig into the methods, it is well described, but also some of them, the methods, for example, kinetic modeling in this case is an in-house script. That's also the case in the other one here. And it makes it very difficult for the reader to know exactly what was done to the data. So this is important since this is what we would like to understand both when we're reading papers, but also when we're dealing with the data afterwards. From the data perspective, the output of a pre-processing pipeline might look something like this. This is an example from output from PVLab, which is a PET pipeline, and this is the output from PET Surfer. And while it produces many files where we can kind of like make sense of what is this, so this is, for example, something with a T1 and something with a segmentation, but it's not quite clear what is the difference between seg2 and seg1. On the other hand, here it also produces a set of files, and we can kind of like infer what is what if we're experts. But for others coming into seeing these files, it's not necessarily clear what is going on. So one of the main purposes of Gitte's and Bob's work as well was to try to make the PET workflows more transparent. So this figure was included, and it basically describes a proposal for how you could capture these different pre-processing steps and also modeling for both the PET data and blood processing, and what kind of meta information would be needed within each of these steps to understand what had happened to the data. And here it's specified for motion correction, registration, some smoothing, delineation of volumes of interest, partial volume correction, but it could also be, you know, other steps, and new steps are being developed all the time. So we of course need to keep up with what is being done and developed out in the field. And the same goes for kinetic modeling and blood data processing. And that leads me on to what we're here to talk about today is PET BIDS derivatives. So this is just an outline of how this may look like. It's an older version, and we'll talk about it more today based on the Google Doc. But the idea is basically that you can try to standardize some of the outputs from pre-processing so they're useful to others to use later on. So what we're really working a lot on in the Open NeuroPET project, for example, is to create these neuroreceptor maps where you have these output files. They're in a given space. You could also have them on the surface. And the goal is to distribute these maps so others can make use of them in a meaningful way. We also produce files like a TSV file here, where you have the time activity curves. So it's the uptake for a given region as a function of time. And the goal is to create pipelines that can deal with this data and produce files or outcomes that are meaningful. So just to give you a short summary, and I'll just do it for the for the different steps here, and then we can talk about specifics after that. So the main template of using PET BIDS derivatives for now is that you have a given pipeline. It could be an open one or a non-open one. It can be your own pipeline. It could be others' pipeline. But the goal is to define what pipeline has been used, and then you keep the same structure as you would have like a sub-dash participant label as a directory. And inside here, you would again have a data type. So that would be an ANAT for anatomical MRIs, but for PET, it would be PET. And then you would have the source entities. Again, that would be sub-01, for example, for a given subject. And then you would have the entities in here, key value pairs, and then you would have a suffix describing this could be PET data, and then you would have some sort of extension describing the file type. And let me just give you an example of how this may look like, as I just described. This could be a pipeline PET surfer. You would have sub-01. You would have the PET directory here, and then inside here you would have sub-01. You would have some information in here in between, depending on what was done to the data, and then you would have _pet.nifty, so that would be the imaging data, and then you would have the corresponding metadata inside the JSON file here. Now, this would be the some outcome data that was established based on the raw data. And one example that is often done to the data as one of the first steps is motion correction. So an output of motion correction could, for example, be that you have, again, this notion, and then you have a description here and -MC to describe that it is motion corrected PET data now that we're dealing with. And that would be your imaging data. And then in the corresponding PET JSON file here, you would have metadata that explains a little bit how this was done. And this is where you guys really come into the picture, and us as a community trying to formalize how this should be done. For here, it could be a reference image, it could be spatial interpolation if that was done, the cost function used, quality control, and then software name or version. For registration, it may look like something like this, where you just define what space it's now defined in. So the rig space would often be the native space that comes out of the scanner. And if you change that now, register to the T1, for example, you have changed the space to T1 weighted instead of a rig. And then you explain that again with a new set of files. This is now the registered PET. And you explain it again with some sort of metadata explaining this process. You can also do this for segmentation, just describing it here a little bit. But the idea is all the time that you have this information in the file name, and if you don't necessarily have it in the file name, you need to have it in the corresponding JSON file. This is how it may look like for partial volume correction, where you could introduce this here. Sorry about this here, but you would have like a key value pair with PVC, and then it's the GTM, geometric transfer matrix that has been used. And again, all the metadata goes into the corresponding JSON file. You can go on, and you can also do this for kinetic modeling, where in this case, we've defined it as a description with kin-mod to define kin-mod, that is kinetic modeling. And then the extension here is that you have, for example, estimated the BP and Ds. This notion deviates a little bit from the example data if you check that out. And there's both some pros and cons with having these different output files, because often you have many parameters associated with a modeling step. So you can either put it into one file, or you can create many different files that contain this information. And then the idea is, again, to have the corresponding metadata in the JSON file. So to keep this presentation short, I just want to really highlight why this is important. I think it's important because we need to join forces to define how this should be defined. It's important because I think we can only do it together. So we make sure that we capture as most of different examples as possible. It will make it easier to understand PET analysis pipelines and what has been done to the data. And in general, it will allow for easier development of reproducible pre-processing workflows. And with that, I want to open up the floor for discussion. So we have plenty of time to discuss. It's 5:20 here in Copenhagen now, so we should have plenty of time to discuss. Yeah.
There's already a question in the chat, and then Nikita, so maybe Jelle can ask his question.
Of course, I muted myself. So what would you recommend for combining pipelines? Because I built a pipeline recently, and it's making use of the fMRI prep and net only pipeline as a start-off point. Yeah, and I added my own extra steps to it. Yeah, so what would you recommend how to implement that in this structure?
Yeah, that's a good question, and very relevant. And I mean, you actually already see it in the MRI community. And if Chris Markowitz or others would like to expand my answer, please go ahead. But there you already see that they have tools for both processing first the MRI data. They also have a tool for doing QC, MRI QC. They do it for fMRI prep for the fMRI data. But the goal is really to, for all these different derivatives here, you could have outputs from the different pipelines. And if they are already there, they can start to interact with each other. And because we now know what are the outputs for example, fMRI prep, we can go in and take that and use it in our own workflow. But it's only possible because we know what kind of file structure the different pipelines they have.
And maybe adding to that, Jelle, maybe just to make this very, very clear that there's no misunderstanding. For example, in the data that Martin has already provided online and gone through, he actually uses the basic FreeSurfer pipeline for the anatomical, and he uses then PET-Surfer for the PET data. So you could, instead of FreeSurfer, just have the fMRI prep and only pipeline, and that would be completely fine. So it's you can completely separate the pipeline for MRI and PET and use whatever is out there, for example.
All right, thank you.
And Nikita, please go ahead.
I hope you can hear me. I'm never sure if my mic works or not.
All good.
Okay, so I was playing with a derivative for PET. And from my experience, it was quite useful to put the output files from different steps into different data type folders. From basic BIDS standard for derivative, you have two standard tags. It's description and label. And if you do one step by on top of the other, these two labels becomes very restrictive. But if you spread the different stages in different data types, like IDIF, I don't know, carotids, modeling, then you resolve a lot of coincidence in the name. It's one thing. Another thing, it's joins a little bit of the previous section. The standard space naming of this derivative, it's extremely restrictive, especially for the PET when it often goes together with MRI. And there, if you have MRI image and PET image, space original, it's a little bit misleading. It's original in which space? So in our laboratory, we use space, PET, and MRI, understanding that there is original kind of subject space in MRI scanner and subject space in PET scanner. And then you can play, for example, reorient the PET image to MRI, and then you have PET image in MRI space. I think it goes against what is written in the standard pre-processing derivatives in BIDS, but I think in case of multimodal data sets, it could be a good thing to implement into the standard. I think it was just two remarks that comes from a little bit naive experience with a BIDS derivative in PET.
Thank you for that comment and feedback, Nikita. I'm just, yeah.
Yeah, I'm also writing some notes into the BEP23 at the very bottom. So please feel free, Nikita, to expand on the points there, so we can bring that up and bring that, for example, up to the steering group as well. And some of them are even here.
And Chris is also writing in the chat, and he's the fMRI prep and other expert in this. And yeah, they use the T1 weighted space to refer to the subject specific space defined by the T1 weighted image. So we could also have it as a space PET to make that completely clear that it's the PET space and not just a rig.
Yeah, so just to say, the standard is supposed to allow you to basically make up any space you want. There are some predefined names that you shouldn't collide with, but otherwise, if you make up your own, then you just need to say, point to what image defines this space.
Mm-hmm.
And there's a spatial reference metadata field for that.
The good thing is that, let me just try to, of course, that there already exists a lot of work on this from the MRI community, is that, of course, and also the other communities, of course, is that we, of course, want to keep this as consistent as possible, what has already been developed. But of course, if we need to, break out of this and introduce our own conventions, this should be, of course, okay. It's just, we want to rely, of course, as much on the others as much as possible.
And Hugo,
okay.
Please go ahead. Yeah.
Thanks for the summary. I was just wondering, I'm not used to derivatives in MRI, but I saw that for PET, you specified that the beginning of the file names are source file. Does it mean that we have to put like a sub, CS, task, or everything that's come from the original image? Because we need to add other information that we have at the beginning in the raw data.
If the data exists, so I mean the BIDS follows the inheritance principle. So if there's only one session, I think sub and session needs to be present, but everything after that does not need to be there if there's only one instance of it. It's more, you could imagine having multiple sessions, and then you need to somehow differentiate between these data types or these type of data. So there you need to include it to be able to make sure that you understand that, this processing belongs to session 1 and this processing belongs to a different session. And if there's anyone, yeah, please, yeah.
Just to add to that, if say you do group processing, the subject entity no longer applies, so you would remove that.
We also, a couple of weeks ago, clarified this in the spec, where we also clarify basically to try to always use the smallest length. So what Martin was saying, right? You have only one run, like say if you do, okay, in PET, except for activation PET, but that's not really what people don't do that much anymore. But you put run 1, run 2, run 3 because you have runs. Otherwise, you don't put it. Session is even not needed either, Martin. If you don't have only one session, no need to put session. So really, your subject could be, for a very simple experiment, very small. But of course, and there is one thing to keep in mind is that you want to limit to 255 characters long anyway. for some operating system issues or some software issues, right? Thanks, Chris. Chris just put in the chat the latest. So yeah, the 255 character long, I came about like same. I was working on derivatives and suddenly it's like, oh, it's too long. If you're not careful, you quickly end up with, sure, the name is there, it's good, you've got all the information, but it's too long. And also, it's not very human friendly anymore, right? When you have a name with so many hyphens and so, yeah. It is something to keep in mind, Martin, that we want to make it still human friendly, readable.
Definitely. I mean, it's always been this trade-off, you know, should it go in the file name or should it go in the JSON file, that type of meta information? And yeah, it's a trade-off.
That's what we put in when you read the doc is really, you put it, and it's Chris who educated me on this because I was asking about something and he was like, why do you do this? And then we decided, okay, let's put it in the spec, which is like really, if it's not needed to differentiate between two files, then it's more a matter of like, do you need it to differentiate two files in your file structure? Right? A bit like same with the space that Nikki was talking about. Like you need to differentiate it, therefore you need two names different. Otherwise, you don't need to. That's kind of like the feel of it.
Mm-hmm.
Although, to be honest, you don't need to for PET to add tracer, but do like to add tracer in the name.
Sure. I mean, we also put it in the JSON file, so it's more. I think of Again,
you shouldn't.
I think there are a few cases where you like use like multiple tracers within the same session, but those are some very rare cases, I think. And again, we try to follow the 80-20 rule, saying that we want to capture 80% of different cases. But of course, we're interested in capturing as much as possible. Just it can be quite difficult with some edge cases.
But then let's maybe move on to Graham.
Yeah. Okay.
Yeah. So I mentioned this in the Google Doc, but I wanted to just bring it up here as well. So one of the things that I think is most exciting with the whole derivatives is being able to like have these modular outputs and then chain them all together, and you have the free server output and then you have this output, and so you can have a blood processing output and you can do all the different things with all your different tools, and each of the different parts talk to each other. So you only need to do the processing once, and say everyone else who wants to use free server can do it in whichever way they need. Is it part of the mandate of the derivative specification to start making some rules about how to define some of the output documents themselves? Say, for instance, I mean, I'm thinking like a tax, a tabular output, where I want to grab the tax from some pipeline for some other tool. Is it part of the mandate to start actually coming up with some rules for how those should be defined? I guess the suffixes can probably be part of that, but I mean, the more standardized we can make the parts that other tools are most likely to use, I think the better can be the chaining of multiple pipelines.
Yeah, I absolutely agree with that. Thank you for that comment. I mean, for me, and I think also the rest here, that is what they want to do. And this is a basic proposal of how it could look like. Again, I'm not sure it's the best, but again, we're trying to agree on something at least, and you know, this is how it could look like for the different regions. And so if we can agree that this is the way that we're gonna output time activity curves, that would be the immediate goal of the PET-BIDS derivatives.
Yeah, I mean, because I'm just thinking if one pipeline does it like this and another one does it in long format and another does it in, I don't know, colored cells and all that. I mean, basically it would be nice to, within the derivatives document, start defining some of the most common outputs that other tools might use and just have like a section for tax should be defined in this way and motion parameter. I don't know, so the kind of common things that might be used as outputs, or blood, for instance, like processed blood.
You can also quickly, make a couple of easy converters. So let's say you have one software which would put a weird format, but this software is quite used a lot. We can easily, take it and make it that it spit out so that it's easy for people to simply, make it change to the format we agree. And then you still have one format only at the end, but we provide the tool that allows you to quickly take that file and then, a little bit what you've done from it with your blood modeling, right?
All right. I mean, I just figure it's like, I feel like at the moment, everyone who develops something is going to be kind of defining their own structure. And I think if we can incorporate a few common kinds of outputs in the definition itself, then we can kind of make it a lot less painless in the future with sort of converters here and there fixing things.
Also, we have to think, one of the things, if we keep saying the goal of derivatives is to also make it more data available for more people, right? Which means if you also want to collaborate with your data scientist friends, which are not from the PET world, it's much easier to provide the tag.tsv than whatever format the given software is spitting out, right? Because tag.tsv, it's easy. And so it's also important to think of reusability for many other people, not necessarily a PET expert, but just thinking out loud of what kind of output is desirable in general.
Mm-hmm.
Yeah, we see there's plenty of other people with their hands up.
Yeah, exactly. Yella, I think you're next.
I was definitely not next, but I think Murat was.
That's okay, but I can go ahead. I wanted to circle back to the file naming and the tags. So in the Open Neuro example, and Martin, you pointed out that your PowerPoint examples differed a little bit, but I'm just looking at the Open NeuroPet example, and it looks like we're already doing some reducing of the file naming. So for example, I noticed that the motion correction tag disappeared when you went into the kinetic modeling stuff, and that makes sense, and that is probably how most people do their processing. So there are certain key pre-processing elements that are assumed to be present in the more downstream tasks, so it makes sense to get rid of those. But for some people, maybe it doesn't make sense to get rid of those because they do want to explore the effect of some upstream pre-processing task on the final outcome. So I think we should give flexibility as to what people choose to keep in the file naming tags. And the other thing here is that I'm seeing that you use the description key with kinmod, whereas in the Open NeuroPet, you invented a new key called KM, I think. I actually like that, the succinctness of KM, and there's a new key invented here, PVC, that's not present in the higher level derivatives description. So I think that can help keep the file names a bit more succinct as well, like using shorter keys that are PET specific.
Sure, I agree with that that notion and those comments. And just to start with this one, for example, and again, Chris, please correct me if I'm wrong, but I think the description key here is that it should be, you should be able to infer, and I think it goes back to this here in the derivatives here, where you would have like a sources where, this file was generated from a previous file, that would be the sources here. But once you have used that file to produce another one, you know, that's the new file. And then you can basically go on, and so you can use a new description, for example, with a file that is being generated further along in this path. And another alternative in this case would be that, I mean, you could also have a, I haven't seen it for the fMRI word, but you could have a motion key. I think usually it's used in the description. And then for the kinetic modeling here, you could also consider to add a model key instead of having it as a description that is more PET specific, but it might interfere a little bit with other specs. So you could have like model SRTM, for example, and then you could remove.
Model would be a bit fraught. Everybody's got models.
Yeah. And then the final thing is that right now it's been used here to explain that this the outcome is, for example, BP&D. This is how it's explained here, but in the example data, it's slightly different here, where you would have the kinetic modeling tag here instead, and then you would have all the output that comes from this model. So you would have like other parameters that are usually estimated for following the modeling. You would have an R1, you would have BP&D, mean squared error, and so forth, that relates to the modeling in one TSV. So you can either have many files that explain each of these outputs here, or you can keep them in, as I've done here, for like having it model specific. But this is something that I would very much like to discuss with all of you. What is your opinion on that?
Are there any comments directly to that, maybe, before we move on to?
I guess I would jump in just a little bit. Is so the desk key or entity came out of the last BIDS derivatives meeting, because essentially we had 5 or 6 different groups all coming up with derivatives and adding entity after entity, and the file names were just going to get extremely long. And so the idea was that the only really, or the general thing that you could do is add in a description, and that's just completely free form. You know, if you want to try to describe your processing in the key or in the file name, you can, but most people are just going to try to say, like, you know, so fMRI just gives you like, this is described as pre-proc. This is the result that you care about of this tool, but each tool decides how they're going to use that field. You can add other entities, but you just need to be judicious about it, because one, there's potential for overlap, and the other is files will tend to accumulate them. And so, yeah, it's going to be hard to decide exactly where to draw that line.
Can I ask something about this? So a bit of new in the whole BIDS nomenclature, especially. But my question is, so now how and when, or what is needed to jump from the use of the keyword description plus whatever, which from what I understand is a more like free text open label, to something that is standard? Because I think we are discussing things now that are most of you is going to use. So I think at some point we will be much easier if we define a basic set of keywords that are always going to be used or mostly going to be used in PET, because at the end that's the whole point, right? Motion correction is something that a lot of people is going to use. So if we created a specific key instead of description, motion correction, etc., it will facilitate. The same for kinetic modeling and partial volume correction. So I think that there are probably 4 or 5 key elements in advanced analysis of PET data that a lot of us will use recurrently And relying on the description, I think we will have the problem that a lot of people will use the description differently and will be counterintuitive, because then everyone will use slightly different, for example, smoothing. Someone will put smooth, someone will put SM, someone will put whatever. So probably there are a few elements like partial volume correction, smoothness, motion correction from kinetic modeling that are probably can become at some point a keyword on its own, but I don't know what is needed to make that, and if that will be more confusing or not.
Thank you for that comment, David. I think that's, and I mean that's also what we have been trying to do for now. If I can just jump into this one here, is that, so I showed it in the slide, some of the keywords, but it's always tricky how you're restricting yourself in terms of allowing only a certain set of key value pairs. So we want to make sure that we capture as much as possible without limiting people from using the tool that they want to use. But I agree with focusing on these, different set of keywords that are used a lot for. You also have the space for registration, PVC, kinetic modeling. Can I think of others? Yeah, I don't know how the rest of the bits come. Please go ahead,
Martin. That's why I was saying we have to keep in mind also the goal of what BIDS is. What is it you're gonna end up is really share. I'm not saying we don't necessarily want to do it. In some cases, you may want to say, well, I do want to share my real-line data, but is anybody gonna ever use your real-line data directly? Or people are more likely to use your final outputs, right? Being the TSV file or being the vertex-wise BND or the voxel-wise UPND. Or really people are going to use intermediate file. My point with that, where I go, is that as we want to store more intermediate file, the longer the name becomes, because obviously, right, it's basically subject 1, step 1, then step 2, then the name of the step and the keywords keeps adding, right? Whereas if you say, well, I'm only interested in sharing what people are going to use, which is basically the final output, or maybe the final pre-processed and then the output after kinetic modeling, right? So you've got your realigned, partial volume corrected, blah, blah, blah. All of this is done. That's the one you share. And then people can do whatever kinetic model they want. And then you also want to share the kinetic models that you did, because, well, why not? Right? And these two are really what people are going to use. Are they going to really use the in-between one? I personally doubt it. Because if you're going to share the real-line data, well, you could also say, well, take the raw and realign it yourself. It's not that realigning is it could be tricky, but, so I think by focusing on the key element you want to share, you also save on how you explain, and of course, everything still needs to be in the JSON, right? I'm not saying don't explain in the JSON. Obviously, you do need to explain. What I'm saying is it solves a little bit that issue of which keywords you need to use and so on, right? Because you could say, just as Chris was saying, for instance, subject 1, description, fMRI prep-proc. Well, you're not describing anything really that fMRI, you're not saying it's been slice time, realigned, normalized, blah, 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 but that's what fMRI prep-proc does. And so in a way, just a sub-desk something, and that's your pre-processed image, kind of solve that issue. Note that we don't want to still define keywords, because if you want to share intermediate file, it's useful. But I'm just highlighting that by think also is like, what do you want to store your realigned images for? I'm gonna reuse those. Right? There's also what is the utility of it. Like, what is the goal? The goal is to share. What do you need to share? And we had that discussion with Robert a long time ago, and he convinced me about it because we were talking about the same thing for EEG, you know, and people were like, oh, we're gonna share something that you filter, you know, like, man, a high pass filter takes no time. Why would you want to store this? It's just compute. And so, you know, I don't know, it's just something to keep in mind. And it solves a little bit of these problems.
Sure, that's
but you know,
I guess we need to get the opinion of the community in terms of, you know, what is often needed. My feeling is that it's quite common to apply, for example, a set of different kinetic models. You often see that in papers, especially for validation papers. You see more region-based analysis than voxel-wise, but if so, the smoothing really comes in. And then I would also say for the groups doing partial volume correction, it's quite common, I would say, to do it both with and without. But here I'd be happy to get others' opinion on that.
maybe it's more like final, you know, when I say final output files, is maybe also the name. Like we could have two sets of name. One which would be where you implement this hierarchy, where you keep adding, you know, key value pairs, versus one where it's just subject description, which refers to all that pre-processing that has been done. It could also be that, you know, it's subject XXX desk, and then the desk is free surfer PVC KMS, you know, like strings, which only because you know what it means, you know, but then it's kind of like you don't have any more of key values, you just have a bunch of values tagged together, and that's your desk.
And maybe just because of the interest of time, Cyril, I think we should, Martin is and me are going to maybe send something out and actually just have a quick poll about this usage. What are people most interested in? I think that's a very good idea, and we can always do that. And Nikita, you had your hand up for a while. You had something adding to this discussion as well?
I would like to slightly disagree with Cyril. That's why I was raising the head. That the advantage of BIDS, it's not only the sharing, but it's also its wonderful through daisy chain, completely different tools. And here you can one pre-processing step doing in one tool, and you can completely append to it something completely different, but because it's all passed by BIDS, everybody can communicate. And here, the all intermediate files becomes, with a lot of keywords, becomes very interesting. For sure, for when you start to publish your results, your pre-processed data sets, you can filter out a lot of intermediary file, but during your processing step, during your pipeline, BIDS make everything much, much easier. And here you need a lot of keywords because you have a lot of much more pre-processing files. It was just my let's say personal opinion.
Thank you, Nikita. I have made the notes. Martin, can maybe scroll down in this document at the very bottom of this document. Now we have just 8 minutes left, so I want to be concise. How are we gonna move forward from here? So thank you everyone already for the ones that have either, you know, commented directly on the doc or now brought stuff up. The plan will now be the following. We will actually try and incorporate and follow up on all the comments that have been made, both in the document, so then you'll get automatically tagged when we reply to your comment, or on the points that were made now. So we'll definitely keep you in the loop. And then the idea is actually, we will send out a follow-up email with the information about this meeting. I know Ronald Pilar and Gaia Rizzo are actually right next door, but at a PhD defense, so, and some other people also couldn't attend, so I'll forward the meeting recording as well as the notes also to those people. And then I think we'll go ahead, Martin, Cyril, and I, and send out a poll in order to get some opinions, and then also to schedule some kind of follow-up meeting after we have an updated version of it. But really, it works quite well to work, I would say, together online. So after we've reached out to you again in about a week's time, please feel free to, you know, keep being pinged in the document and responding and having the discussions there, because then everyone can see what's going on.
Yeah, thank you for that, Melanie. And just summarizing on this, it seems like there is indeed a lot of focus, both of course, naming and output files. What would be really useful is that based on this information now, depending on the use case that you have at your imaging center with your PET data, if you could like try to convert some of your data into this structure, because it gives a really good sense of what does it cover, what it should, or do we need to add something to cover what is going on. And we can of course only do it for many of the examples that we can think of, but it would be really useful if you could try to do that for your own data as well. And then
and maybe Martin just adding there, and you don't actually have to convert it. It's totally enough for us if you just take your experiment and make a theoretical thinking. If I would want to write that down in a BIDS PET derivative file name, how would that look like? So we don't actually want you to go through the trouble of renaming files and everything on disk. It's totally enough if you think through the experiment and then say, okay, the name would end up looking like this, and then add that name for us, please, so we see where the issues and problems are.
Exactly. And one of the main things that we need to do now trying to develop this this standard is that we need to have a lot of use examples. We also did that for the BIDS raw. So examples that you come up with, they will be incorporated into the standard. What I would really like is also that, you know, there's obviously some different cores that people are experts in. Mine really comes from the pre-processing, so that's right up until basically I'll also do the kinetic modeling, but also blood data processing. But I'm very interested in, again, you know, trying to define files that, for example, Granville can use for KinFitter. So if he knows that I have an output file that, you know, this is the output from pre-processing, this is how it looks like for tags. But you also have for the imaging data, both on surface and volume. These are the output files. He can take that and use it for the modeling. But it would also be useful for those that are interested in modeling to try to define what kind of metadata and how should the output of model data look like. So it would be nice if people could, depending on what they're interested in, try to focus on especially files within those different domains. So pre-processing, blood data processing, and kinetic modeling.
Yeah, Richard, please.
Just a quick comment. Just in my experience, as we develop new methods, we're never going to get all the parameters right for the things that we'll be doing in the future. So if there is a way to anticipate that, that doesn't mean we have to keep, we're not going to keep updating those fields, but if there's a way to add additional ancillary information that is just not in the format, so at least there's a paper trail of everything that we're doing. We use weighting functions for how we weight our data. We have that vector of weights that's going to be different from everybody else's. We have that information to include. We don't necessarily want that to be a field for everybody. And that may let us grow in addition to say, oh, I want to add that too, but we can save all that information. That may allow this to start a little smaller with the key features that everybody needs, and then slowly say, oh, we should be adding this as a fixed field because it's being used by lots of people. That way we grow.
Thank you for that comment, Rich.
Great, then there are a few more comments in the chat and I will follow up on those and transfer them. But otherwise, it's 2 minutes before. I know, especially here in Europe, everyone is on the way out. It's getting dark in Scandinavia outside by now. Thank you all so much for coming and making this you know, a constructive way forward and helping us to push this somehow. Please feel free to reach out. I will save the chat and I will also save the recording, and then Martin, Cyril, and I will reach out to you within the next week with some updates. And yes, the to-dos are basically, please look at the Google Doc if you have time and try and make a thought experiment about how you can name your experiment and get something meaningful out of it, so we discover the holes in our thought process so far. So thanks everyone. Have a very nice weekend and we will be in touch.
Bye all. Thank you.
Bye.